Okay, so hi, my name is Jocelyn. For this project, I'm explaining a protein called pisa 1. For my audience, I decided to explain this to my mom, who is not a part of the science or medicine. So I'm just going to try to keep things very simple and related to everyday ideas just to better her understanding overall. So I'm going to start introducing my protein, pisa 1, which is found in the cell membrane, which is the outer layer of a cell that controls what goes in and out. Piezo 1 acts like a mechanical sensor. This means that it can detect physical pressure or stretching in the cell membrane. And when the membrane stretches, piezo 1 opens and it forms a channel that lets ions like calcium move into the cell. So these ions help cells send signals and respond to physical forces. Overall, pisa 1 is very important in several types of cells, especially red blood cells and cells in our blood vessels. Um, and then when this protein doesn't work correctly because of a mutation, it can cause conditions like hereditary serocytosis, which this disease can affect the red blood cells and cause them to lose too much water, which can make them dehydrated and not function properly. So now I'm going to show my model, which I'm going to attach the video to this video. But overall, the overall shape of the protein looks like a three-bladed propeller or a fan sitting in the cell membrane, as you can tell. In my model, the center hole represents the ion channel, which is a pathway where the ions move through the protein and into the cell. The curved arms around the center represent the different parts of the protein that respond when the membrane is stretched. So now I'm going to move on to the explaining the different protein structure levels. So as you can tell, the proteins do have different levels, which I represented all of those different parts into my model. First is the primary structure, which is a sequence of amino acids that make up the protein, and overall the amino acids are the building block of proteins. And in my model, the colored beads represent the amino acid chain. Now the secondary structure, which refers to the small shape formed by the chain. These shapes include alpha helices, which look like coils, and beta sheets, which look like folded zigzag sections. And for these, I used pipe cleaners shaped into coils and folded these to represent these different structures. And then I have the tertiary structure, which is a full three-dimensional folding of one protein chain. As you can tell in my model, the curved arms show how the protein folds into its overall shape. And then finally, we have the quaternary structure, which is when the multiple protein parts come together to form one functional protein. But overall, pisa 1 forms a very large structure made of several interacting parts, which together they create the channel in the center, as you can tell. The mutation, which is at the very bottom in this model, it shows that it means that one amino acid in the protein sequence has changed, and as you can tell, I labeled it. But normally amino acids interact with each other through different chemical interactions, such as hydrogen bonds, ionic interactions, or hydrophobic interactions, which help the protein fold correctly. And when one amino acid changes, it can affect those interactions and slightly change the protein shape. And how does a mutation affect the protein? Well, in pisa 1, certain mutations cause the protein to have more activity than normal, meaning the ion channel opens more easily or stays open longer than it should. And this allows too many ions to enter the cell, which disrupt the overall balance of water and salts inside the cell. And in red blood cells, these imbalances cause the water to leave the cell, which leads to dehydrated cells. This is why many people with hereditary serocytosis have red blood cells that don't function normally. And at the level of the whole body, this can lead to different problems like anemia, fatigue, and issues with oxygen transport. And then finally, the limitations of my model. I first think the size and the scale are not accurate at all because the real proteins are extremely small, much more complex in this model. The second is that the protein in real life is constantly moving and changing shape while my model is completely static. And then third, my model does not show all the chemical interactions such as hydrogen bonds and electrode charges that help maintain the protein structure. But overall, it's like really helpful to understand the basic idea and how much a protein is much more detailed and dynamic. But overall, this is my protein, and it overall just helps cells respond to physical forces. And I really hope you enjoyed learning and listening to my explanation of pisa 1.